Well, thank you. I just want to say I'm happy to be here. I just retired from ARS just before I came to Australia, so I'm doing this in my retirement, but which you may or may not think is a very good use. But I'm going to talk about multiple things today. First, I'm going to talk about the highlights of the Global Herbicide Resistance Challenge. This was held 2 weeks ago in Perth. We had over 300 attendees with representatives from approximately 30 countries. And we spent 4 days talking about all different aspects of herbicide resistance. And I'm going to try to hit what I think are some of the highlights. And then I'm going to talk about 3 other topics of which I've done research in recent years. One is the development of a field kit for glyphosate resistance detection. Second is herbicide crop residue interactions. And finally, a phenomenon that I don't know whether you have it here or not, but it's accelerated atrazine degradation in the soil. So the Global Herbicide Resistance Challenge, what were the main take-home messages from that? Well, one is that herbicide-resistant weeds continue to increase on a global scale, which back in 1995, I wrote a paper on this, and at that time there were 82 species that had resistance, and today in 2012 there's over 200 species that have resistance, so we really haven't managed the problem very well. The other concerning thing was multiple resistant weeds are rapidly increasing, not only here in Australia, but also in the United States, where farmers have used up a herbicide, such as a Group A herbicide, moved to a Group B, selected for resistance to that, moved on to glyphosate, and have managed to select for resistance to that. The other thing is that there's both target site, meaning mutations at the actually target site of the herbicide, and non-target site mechanisms. The non-target site mechanisms tend to be metabolism-based. The target site resistance comes from high use rates, and usually just one gene controls that. The non-target sites are from using multiple years of low rates, so you're kind of damned if you do, damned if you don't. But the problem with this is that it's becoming increasingly difficult to find herbicide options in some of our crops because we've selected for so much resistance on certain weed species. One of the things that really came out, and something that you here should be concerned about, is the explosion of glyphosate-resistant weeds in the United States and what impact that is having on farming. Palmer amaranth is a very pernicious species. It's got multiple resistance to Group A, Group B, Group C, paraquat, a lot of different resistances. And in Roundup Ready cotton and soybeans in the South, the farmers were using multiple applications of glyphosate year after year with no other herbicides in the mix. So they were doing the worst case scenario that if you wanted to try to get resistance, they were doing it, which was 5 to 6 applications of glyphosate per year with no other herbicide. And up until 2006, they were getting excellent weed control. And then in 2007, they found a certain population. It happened first in Georgia and then spread throughout the southern U.S. Of Palmer amaranth that wasn't being controlled. And initially it was an isolated field, and then it was counties, and now it's multi-state. And farmers today in 2012, in order to manage this one weed, which is a driver weed and is driving them out of production, are using up to 7 herbicide applications with multiple mechanisms of actions, plus hand hoeing. And initially they're paying up to $300 or over $300 per hectare to hand hoe their cotton in order to be able to control the weeds. And so this happened in about a 5-year period. The other case is kochia, which I don't think you have here, but it's a tumbleweed. It's a very bad weed that we have in the western United States under dryland conditions. And it was also being controlled primarily in fallow with multiple applications of glyphosate. They found their first case of resistance in 2007, and now 5 years later, it's widespread throughout the Great Plains, stretching from Oklahoma to Canada. This is a picture, 2 pictures of a cotton field in 2006 or 2007 when there was just random Palmer amaranth plants in the field. And farmers, when they first saw this, their answer to that was just increase the rate of Roundup or increase the frequency of Roundup application. If you look at the other picture, that's where they are today. they have very, very high populations of glyphosate-resistant palmer amaranth, and essentially have had, in many cases, if they don't do all this very elaborate procedures, essentially have to abandon the field because they cannot control the weed and they cannot harvest the crop. The bottom two pictures show kochia. As I said, it was a tumbleweed, and this is what was first seen, where this mother plant tumbled across a fallow field. If you could see more closely, you would see that there are dead plants out there, but where this mother plant that was glyphosate resistant tumbled across the field shows up very, very plainly. And then the other picture on your right shows that this is what they're facing in certain circumstances. And so this tells you how fast a system can break. Glyphosate resistant crops were the answer to herbicide management or weed management in the United States beginning in 1995. And we had about 10 or 15 years of good, excellent, simple weed control, and now the system is breaking. And part of the problem is that farmers were only applying glyphosate with no pre-emergence or very few other post-emergence compounds in those programs, and they're becoming an increasingly greater problem, and they're having to go to more complex and more expensive systems to manage it. They also talked about new cases of resistance in Australia, 2 species of glyphosate-resistant bromus, resistant bed straw, group A resistant Phalaris paradoxa and minor, and group C and F resistant Indian hedge mustard are some of the cases that they highlighted. So those are the problems. What are we doing to answer those problems? Well, the new technologies that were discussed was in the United States, and I assume eventually it may come here to Australia, is stacking herbicide resistance in the crop. Putting together glyphosate plus 2,4-D resistance in things like cotton and soybeans, as well as glyphosate plus dicamba or glyphosate plus glufosinate, with the idea that the additional partner, these are stacked in the same species or same variety, that they'll be able to use 2,4-D dicamba glufosinate to control these glyphosate-resistant weeds. The problem that faces is that with the kochia, for example, we already have dual resistance in populations of glyphosate plus dicamba, because we had selected for dicamba resistance earlier in our fallowing situations. And this system, if it's not used right, is going to break just as fast or faster than we had with the glyphosate. Essentially, like the glyphosate plus glufosinate, farmers are using this in the south to try to control their glyphosate-resistant weeds using the glufosinate, and it's doing a pretty good job if you apply it early and have the right conditions. The problem is some of the farmers are doing the same thing they did with glyphosate. They just switched to glufosinate, and that's a recipe for disaster. But there are responses. One of the other things that was said at the meeting was the new decision support software, the relaunch of RIM, a weed seed wizard, and information that you have in your update on WeedSmart and WeedSmart applications, which allows you to look at different rotations, crops, herbicides, and to get an estimation of how good a resistance management procedure that is. It's a fairly simple system. There's an app that you can download onto your smartphones or iPad that allows you to quickly look at different scenarios to see whether your program is good or bad. Probably the thing that caused the most excitement at the meeting was a new technology that was introduced from Monsanto called BioDirect. This is a brand new method as far as herbicides go to manage weeds. It's what's called RNAi-based technology. What this technology does is essentially spray on small double-stranded ribonucleic acid onto the plant, which is taken up by the plant, and essentially silences certain genes. In this case, they've been able to reverse glyphosate resistance by going in and shutting off the target site, which is EPSP synthase, and reducing the amount of that in the plant and spraying that along with glyphosate to give them control of palmer amaranth, for example. It's also been shown to work with other modes of action such as HPPD, PPO, and I know they're also looking at ALS inhibitors. Right now, this technology is in its infancy. They've had a proof of concept in the field. It's just starting and it's years before it'll be developed, but if this technology works out, it promises to give us a new tool that is very, very specific. That's one of its positives, is it's very specific. It's actually species specific because the RNA sequences are specific to species. So that kind of the weakness is you have to have various ones. If you're trying to control a broad spectrum of weeds, you'd have to have multiple forms of this RNA in this tank mix. But I think it opens up a whole new area that we can start looking for new mechanisms of action to help us combat herbicide resistance as well as new weed management tools. The other thing they talked about, and I think you'll get more talk about this, or much more detail, is weed seed capture and destruction, and the test by GRDC of different methods for weed capture, and I'll not spend any time on that. So those are, to me, were the highlights from the meetings, that herbicide resistance is increasing, multiple resistance is increasing, and the need for diversity in weed management programs. That if you are depending on a single technology, taking out pre-emergence herbicides out of your system and just basing your whole program on post is not going to work in the long run, and that you need to think of your program, and integrated weed management is the only way to go. So shifting gears, I'm going to talk now about something that I've worked on in the last year or so, and that's trying to develop a field kit to detect glyphosate resistance. Right now, if you have a suspected glyphosate-resistant population, you usually collect seeds, take those seeds into the greenhouse, spray them, grow the seeds up to the plants, spray them either with multiple rates or discriminating dose, determine whether or not you have resistance, and if you do, then look to see what the resistance is. This is a very robust system and works very well, but it takes a long time. And it takes a lot of resources. And so one of the things that I was working with Monsanto is they have developed a new method to detect shikimate, which is a metabolite, and I'll talk about that accumulates in plants. It could potentially be done in the field. So this is how glyphosate works. It works on the aromatic amino acid biosynthetic pathway. You can see that the target site is EPSP synthase. This is what glyphosate inhibits. But one of the consequences of that inhibition is the accumulation of a metabolite called shikimate. And the amount of shikimate accumulation is related to the efficacy of the herbicide. So that if you have a susceptible plant, you spray it with glyphosate, it accumulates very, very high levels of shikimate. On the other hand, if you have a resistant plant, the resistant plants only accumulate shikimate at very, very high levels of glyphosate, because you know most of glyphosate resistance is anywhere from 5 to 10-fold. And you can use this difference between susceptible and resistant plants to screen for resistant biotypes. This is an example of Lolium rigidum, glyphosate-resistant Lolium rigidum. What you're looking at here on the y-axis is shikimate accumulation, and on the x-axis is different concentrations of glyphosate. The blue line shows shikimate accumulation in a susceptible biotype, and you can see that it accumulates fairly rapidly, where the red line is the resistant biotype. This particular biotype is a uptake mutant or translocation-based mutant. So at low concentrations, around 10 micromolar, you'll see that there's a clear difference between the resistant and susceptible biotype on glyphosate or on shikimate accumulation, where at high concentrations, this difference disappears. And we utilize these differences in order to screen for resistance. And what Monsanto has done is they developed a enzyme system that essentially takes shikimate and converts it, or takes, shikimate and converts it to 3-dihydroshikimate, but the thing that it does is it produces a reductant, NADPH, which can then be used to take a dye that is colorless. When it's reduced, it produces a color. And so what you get is this type of situation. If you look on your right or on your left, you'll see different rates of shikimate, or on your right, I guess. This is the kind of, so as you increase shikimate accumulation, you see a deeper and deeper color, and this is what the spectrophotometer would see. And so using this type of procedure, it gives you a colorimetric assay that you can see by eye in the field, and allows you to look at the levels of shikimate in the plant to determine whether or not it's resistant. And so the obstacles I had to overcome is how stable was the assay, ways to extract the plants, whether to screen whole plants or leaf disc, glyphosate concentrations, and interpretation. And so what we've done is we've developed a system where you go out in the field and you take leaf disc or growing points from plants that you suspect or may not be glyphosate resistant, and you expose it to 3 different rates of glyphosate, 0, 100, and 10 millimolar. That's not really important, but what we have found is that by using these 3 rates, we can differentiate, and I'll show you the difference between a susceptible and resistant biotype. And we found this works on a number of species. We've shown that it'll work on fleabane, on Palmer's amaranth, on water hemp, giant ragweed, kochia, sourgrass, and from your point of view, ryegrass as well. Those are only species we've looked at so far. This is what the kit looks like. You get a punch that allows you to take a leaf disc. You get various solutions here that you put these leaf discs in, A, B, C, and T, and then you incubate this under light for 24 to 48 hours, freeze thaw this a couple of times, take a dropper of that, add water to this, which has the enzyme solution in, and then let it, in 10 minutes you know whether you got resistance. I've used a portable light box. You can do this under a lamp on the desk, but you need to have light in order to work. And these are the kinds of results that you would get from this kit. So this is a control. So this is a blank that shows no shikimate accumulation. This is a susceptible species, and this is a resistant species. And what you're looking at is the difference here. This is a very high concentration of glyphosate. That's in there just to tell you that you picked the right tissue, so that the system works, shikimate is accumulated with glyphosate. And what you're really looking at is this. Does this look like this or like that? If it looks like this, then it's a resistant biotype. If it looks like that, then it's a susceptible biotype. So it's a fairly simple yes-no answer. Do I have glyphosate resistance or not? So far, this still is in development. I've sent this kit to various researchers throughout the U.S., and there's still some questions about how to interpret the results. Because it's a biological system, it depends on taking tissue from actively growing plants. And the next step is to find someone to produce the kits and test them more widely. So this is, again, kind of in its infancy, but it's a possibility that you could use in the future. Now, where would you use a kit like this? Where you would use a kit like this is, if you remember back the picture I had of a 1 or 2 Palmer amaranths, or if you have a paddock that has just spots of weeds growing in various parts of the field, and you're not sure, well, was that a skip because I sprayed Roundup, or is that resistance? This type of kit would allow you to look at that and determine, well, yeah, it's not resistant, so I must have had a skip or some other problem with my application, or it is resistant and I better change my program right away in order to do that. Once you have a disaster, this kit is immaterial. If you spray glyphosate and the whole field's weedy, infested, like you saw in our that second picture I had of the Palmer amaranth, you don't need to ask a question there, you already know you have resistance. So this type of kit, in my mind, is early on. One of the things that was emphasized at the Global Herbicide Challenge as a management tool was to scout your fields and to look for resistance, look for the unexpected. And this type of kit would give you a quick way to look for and determine, do I have resistance or do I have something else? Now moving on to the third topic, and I'll take questions at the end of this, is one of the things that John asked me to consider is what is the effect of crop residue, particularly as we go to more and more pre-emergence herbicides to manage herbicide resistance. What is the effect of crop residue on herbicide efficacy of soil-applied herbicides? Because we know one goal of conservation tillage is to preserve the crop residue, because it preserves moisture, but it can also be a major barrier to soil-applied herbicides. So we conducted studies in the laboratory to compare the interactions or the effect of residue on atrazine, metolachlorine, and a newer herbicide, peroxysulfone. Now these were laboratory studies. They weren't done under under real-world conditions. We used very high spray volumes, 400 liters per hectare, because we wanted to get uniform coverage. And we use fairly small droplet size. We just had a discussion on that. And so that may or may not affect it, although I have some results that would suggest that what we found in the greenhouse or in the laboratory is what's happening in the field as well. So we used peroxysulfone or Sakura. It's a relatively new soil-applied herbicide that's developed or sold here by Bayer. It's a Group K herbicide, and can be used at 118 grams or used at 118 grams per hectare. And the label says it can be used in up to 50% residue cover. So we wanted to see, well, what is the effective residue cover on terms of both interception and what, if you have rainfall after that, will happen? How easily is it washed off the crop residue? So we did this study on wheat, corn, and sorghum residues to look at see if there's any difference between residue. And first we just determined how much of these herbicides, if you have them in a system where you have the residue and the herbicide by itself, how much binding is there occurring? And then if you spray it through a residue, how much is intercepted, and then simulated rainfall after that, how much is washed off, and how much may be actually lost from the residue if it's left on there dry. So this is what we found in terms of binding, comparing these three herbicides, peroxysulfone, metolachlor, and atrazine. There's a lot of data here, but the thing is that there's differences. If you look at here like peroxysulfone, whether in sorghum, whether it was the stem, the head, or the leaves, there was a difference in how much herbicide bound. So the most binding in all these cases, or in most of these cases, was to the stem. If you looked at the head, the remains of the head, or the leaves, for peroxysulfone there was a decrease. For atrazine there was a decrease. For metolachlor it didn't really make any difference. If we looked at wheat, we only had stem. We can see that, again, peroxysulfone of all these bound the least, followed by atrazine, and then metolachlor was bound the most. And then if we looked at maize, again, looking at the stem, cob, and leaves, similar to what we found with the sorghum, that the herbicide peroxysulfone tended to bind more to the stem than it did to the cob or the leaves. It bound very, very poorly to these. Metolachlor didn't make a lot of difference, and atrazine, again, we found more bound to the stem and less to the cob or to the leaves of the species. Now this is just looking at what we call a batch slurry, where we took 1 gram of plant material, put it in water, 10 mils of water, and then we added the herbicide in the water and we shook it for 24 hours, and then we determined how much was left in the water. This was a relationship we found between crop residue cover. Now I'm not looking at tons of residue per hectare, I'm looking at total coverage on the ground. And this is looking at residue that's lying flat on the ground, not necessarily standing up. And we found a straight line relationship is that as the percent ground cover increased, there was an increase. This is percent intercepted versus ground cover. There was an increase in ground cover. But you'll notice that even with 90% ground cover, the maximum we got was about 60% intercepted. So there's not, even though it's a straight line relationship, the amount that actually got through the ground cover is somewhat varied depending on, well, it was less than the actual percent cover. But it does show that with increasing ground cover, you get increasing interception. So how fast does this come off, and how easily does it come off? Again, looking at the three herbicides, this was from sorghum residue, and in this case, we had the herbicide, sprayed it over the residue, determined how much was intercepted on the residue, and then we let the residue sit in the greenhouse on dry for 6 days, and then we applied the simulated rainfall of 5, 10, or 20 millimeters. And what you can see is that even with 5 millimeters of what was on the residue, we washed off between 60 and 70% of the residue. And again, peroxysulfone, there was no significant difference between these. As we increased spray volume or increased rainfall, there was an increase in release. So like the 120 millimeters of rainfall that you got this week, if you had any herbicide on the residue, most of it would have sprayed or would have been washed off. But there is a certain amount, and the same thing we found with wheat residue, very similar to corn residue, is that in this case, because it binds a little more tightly, you see a little more less release with the 5 millimeters, but at the time you got up to 20 millimeters, you released about anywhere from 70 to 85% of the herbicide. So it's saying that the herbicide that's intercepted by this residue can be released. Although there is we found anywhere from 16 to 18% was not released. No matter how much rainfall you put on it, you're not going to get it off. It was bound irreversibly. The other interesting phenomenon we found was how much residue stayed on those plants in that 6-day time period. In the case of peroxysulfone and atrazine, we got 100% back. So we determined how much was on there and then how much we got after 6 days. Interestingly, with metolachlor, we found that we lost about 20 to 40% of our sorghum residue. We lost about 40% of the metolachlor, either through volatilization or some sort of degradation process during that 6-day period. If we looked at wheat residue, we found similar things, although it's not as great. What's interesting down here, when we looked at we had some wheat residue. You see that's blanked. And what that was is my student went out and collected some wheat residue from a farmer's field. Of course, he didn't ask the farmer, so we don't know if we can go back to the farmer and ask him what he did. But obviously, the farmer had sprayed atrazine on the field because the wheat residue that he collected had high concentrations of atrazine on the residue. And this, it looked like it was very similar to what we found when we sprayed 1 kilo per hectare of atrazine in our test system. So that tells me that what we saw in our test system in the laboratory is also occurring in the field. But we also found that when we did the simulated rainfall, that herbicide released, even though I have no idea how long it had been on there, but it had been some period of time. So, just a summary of that, of what we found, is the order of binding to the crop residues was metolachlor greater than atrazine, greater than peroxysulfone, but all the herbicides are washed off the residue by simulated rainfall. 20 millimeters of rain washed approximately 80% from the residue. And in our case, peroxysulfone and atrazine were very stable on the crop residue over a 6-day period, where metolachlor appeared to be lost, possibly through volatilization or some other process. Now, switching to my last topic, and I know I'm covering a lot of kind of the waterfront here, is some work we've been doing on accelerated atrazine degradation. I know you use atrazine and simazine here to manage weeds in sorghum, maize, sugarcane, lupins, and TT canola. And atrazine is generally considered a long residual herbicide. If you look at in CropLife Australia document, atrazine has a median half-life of 41 days in Australia, depending on microbial population, soil moisture levels, soil temperature, soil pH, and farming practices. And this has been the accepted paradigm, or what if somebody tells, well, how long does atrazine last in the soil? And the answer is, well, it has a half-life, meaning the time it takes for 50% of it to degrade, between 40 and 60 days. And that's what I thought. And so, and if you looked at the old literature, you find that atrazine is considered to be, for many years, primarily degraded by chemical hydrolysis, or I should say detoxified. It can also be metabolized by microorganisms, but the metabolites that are produced by the microorganisms are still phytotoxic and still readily available. It's only a cleavage of the chlorine ring or chlorine substituent on the triazine ring that really detoxifies. The other thing is that it's always been said that atrazine persists longer in high pH soils compared to acidic soils. And that is because this chemical hydrolysis process works better under acid conditions and basic conditions. So it's considered a long residual soil-applied herbicide, and it has a potential to leach due to this long life, allowing movement to the groundwater. But what we have found, I stumbled on this in Colorado when I was trying to do something completely different, but I was looking to see the movement of atrazine under different irrigation regimes, and finding the atrazine was disappearing much more rapidly than expected. And we found that atrazine the soil microorganisms have adapted to glyphosate, so that they now can degrade atrazine very, very rapidly. and that there are certain genes, there are 6 different genes that are contained on a plasmid, which is a small circular piece of DNA that bacteria use to exchange genetic information. These genes that encode for the enzymes that degrade atrazine can be found throughout the world. They've been found in Europe, in America, Canada, and other places where they have a history of atrazine use. And in these circumstances, atrazine becomes a very short residual soil-applied herbicide. The upside of that, it doesn't tend to leach in those circumstances because the half-life, the rapid degradation, just doesn't give it time. And I'll show you some data on that. So this is the old paradigm of how atrazine was degraded. And that is the microorganisms would either, this is atrazine, and the microorganisms would either take off the isopropyl or the ethyl group. But these two compounds are still phytotoxic. And they're fairly, in fact, if you look at groundwater data, you would actually in many cases find higher levels of these two metabolites than the parent compound atrazine. This is the real true degradation pathway or detoxification, where you take this chlorine group and you hydroxylate it to a hydroxy. So this hydroxyatrazine is very poor herbicidal activity, plus it's bound very, very tightly to the soil. But this is a new method, or new pathway that we found in these soils with this accelerated atrazine degradation. And what's interesting is the first step is this dechlorination or hydroxylation. So in these soils that you find this enhanced degradation, you see very, very low levels of the des-isopropyl or des-ethyl. And what you see are transitorily high levels of hydroxyatrazine, but then that is very rapidly metabolized, eventually to CO2 and ammonia. So it completely degrades the herbicide. And what the microorganisms are doing, they're using it as a nitrogen source. And this is what happens in the field. And this is a real world situation where we were looking at a farmer's field where he had sprayed atrazine at 1 kilo per hectare on April 30th. And we followed the rate of the level of the herbicide in the field, and you can see it degraded very rapidly. He put another application on May 30th, and this dissipated even more rapidly, so that by the middle of June, there was no atrazine left in that field. And under these circumstances, it wasn't Roundup ready, and so he didn't have anything to manage his weeds, and he had a disaster on his hands. And so we looked at a number of fields in eastern Colorado. All of these had a history of atrazine use of 5 years or more. In every case, atrazine is dissipating very, very rapidly in these fields in eastern Colorado. And in fact, when we looked at these fields over multiple years, we found half-lives of atrazine from about 7 days down to 3.5 days. So this herbicide is dissipating very, very rapidly under these circumstances. And this is the kind of situation that farmer I was telling you about that had the atrazine, and he was dependent on atrazine to control his weeds. And you can see that this is after the second application, well, after the second application that the herbicide was not acting, and by the end of the season he had a complete disaster. As I said, this accelerated degradation is due to a change in the soil microorganisms. They can share this information across species, and if you can extract DNA from soil and actually look for the presence of these genes in the soil. And in our circumstances, we've done that. We've looked for this band here is the level of this particular enzyme, which is an indication of how high this microorganism is existing in the soil. And if you go from very few or minimal applications of atrazine, you can see it's very low. And if you had more than 4 years of atrazine use, you have very, very high levels of this enzyme, indicating that you're enriching the soil for this particular microorganism. We found that the factors, we've done some work, and then we also, this is a model based on 90 soils from throughout the world, and a review that we wrote, or that a colleague of mine wrote, and then also some work that I did with other colleagues at CSU, that the main factors that are controlling atrazine degradation is soil pH and years of use, and whether or not atrazine was used the year before. And when I say pH, what I mean is that this phenomenon, unlike the old paradigm where atrazine degrades more slowly and is more persistent in acidic or basic soils, In the case of this accelerated atrazine degradation, it's just the opposite. If you have soils with pH greater than 6, then it will degrade very fast, very rapidly if you have these bugs available. If you have pH less than 6, then you go back to the old system and the herbicide persists much longer. And this, out of this using this atrazine degradation model, in this review that we wrote, we looked at where is there potential for glyphosate enhanced degradation to occur throughout the world. And you can see that it's the blue area. There's very few areas where you wouldn't expect it to be happening because of the atrazine use pattern as well as soil pH. And this was some work that's been done here in Australia, looking at atrazine degradation and transport in a black vertisol soil. And you can see that the atrazine in this case may not be dissipating as rapidly, but if you actually look at this graph here, this is actually what's atrazine is bioavailable, and you can see that it's dissipating. The half-life is not 40 days. The half-life is more like 10 days. 8 days. So this phenomenon has been found, and I'll be interested from feedback from you if you have noticed that atrazine doesn't seem to be working quite as long as it used to. It's possible that you have this enhanced atrazine degradation. And this is what effect it has on environmental fate, where we're looking at leaching of the herbicide. We're looking at the top 30 centimeters, and where we have the enhanced degradation, you can see that the herbicide never moved below 15 centimeters, whereas in a soil that did not have the enhanced degradation, we saw tremendous movement of it down the profile. And so where you have this enhanced degradation, you have much less leaching of the compound. So accelerated atrazine degradation occurs worldwide. I've tested soils throughout the US, Hawaii, and they've also been, a lot of work's been done in Europe and other areas showing this phenomenon. Where the herbicide has been intensely used, the soil microorganisms can often metabolize. With accelerated degradation, the persistence will be greatly shortened, but there's also less leaching. One of the phenomenon, when I was working with a farmer in eastern Colorado, I told him he had this enhanced degradation, and he said, that's fantastic. Because the way he used atrazine, he would apply atrazine early post on his corn to get the pre and post-emergence activity, and then he would spray atrazine again at lay-by, along with metolachlor and Roundup. And then and he had very good weed control, because where you have this accelerated degradation, instead of getting 6-8 more weeks of atrazine efficacy, you'll get more like 3 weeks. But he had tremendous weed control, and then he was able to plant annual rye on his field as a cover crop for animal production and had no problems with carryover. And I'm not advocating that, but last year I had a bunch of safflower growers who saw this work, and they had sprayed atrazine on their corn the year before. They wanted to plant safflowers, and they wanted to know if they could, because the label says you have to wait 18 months. But where they had this accelerated degradation, there was no atrazine left in the soil, so they could safely plant their safflower. So that's all I have. Do I have any time left for questions?